Hi, I'm Giovanna and I'm going to present this work about the description of a new species of Macrothrix which was financed by FAPESP. I'm a professor at Unesp Registro. Daniela Freitas is a student who received a FAPESP grant for this proposal, and Gilmar PERBICHE-NEVES and Diogo SOUSA were the specialized researchers who made this study possible. This study begins with a question. How is the knowledge of the Macrothrix genus in the world? The publication about Macrothricidae have been used for almost the last 3 decades as a basis for improving our taxonomic knowledge of the Macrothrix genus, and drives the development of the taxonomy, biogeography, and phylogeny of this group. With this, it's possible to indicate the consolidation of stable groups of species within 4 Macrothrix groups, with we can see in these figures. However, for many species of Macrothrix, an adequate description of important morphological traits is still lacking. Macrothrix marthae bears a combination of morphological traits which are related to the limbs, which indicates its epiphytic habit that does not fit in any species group. Therefore, this study aims to describe a new species of Macrothricidae for São Paulo state in Brazil that shares some morphological traits with M. marthae. The study area comprises the Ribeira de Iguape River basin, located in the south of São Paulo state. The samples were made in Valo Grande, an artificial channel of communication with the sea, near the urban core of the city of Iguape. The samples were collected on the margins and at the center of the river monthly during a year, with a plankton net that was horizontally dragged on the subsurface of water for 10 minutes. The obtained material was fixed in absolute. In laboratory, specimens were selected under a stereo microscope. The morphology of the limbs and the other structures were studied with a microscope. The description style followed the Van Damme 2016, which separates the anatomic structures into blocks: general habitus, carapace, cephalic, thoracic limbs, abdominal, and post-abdominal. The new species of Branchiopoda, Macrothrix lourdesae, was named in honor to the Dr. Lourdes Loureiro for her important contribution to ecology, biogeography, and taxonomy of Cladocera in Brazil and South America. Several morphological traits might be used to distinguish Macrothrix lourdesae from other species, namely, on the ventral margin of head there is a protuberance near antennally base. This morphology is similar in M. marthae. For the other hand, other species of Macrothricidae do not bear protuberance on the ventral margin of the head. The most consistent set of morphological traits that separate M. marthae and M. lourdesae in different species can be observed in the limbs. In the first and third limb, the setae are heavily modified for the epiphytic habit. However, they differ one from another because M. lourdesae has 3 setae on the exopodite of third limb, while M. marthae bears only 2. The fourth limb is reduced without exopodite and without setae on the basal endite. The fifth limb is extremely reduced. The last morphological trait is related to the post-abdomen, which M. lourdesae has a shorter ventral margin when compared to M. marthae. The post-abdominal claw in marthae-group species is markedly shorter than the claws observed in other species groups within Macrothrix. A careful study on the morphology of males and epiphyal females of M. marthae and M. lourdesae is still lacking. However, our findings based on parthenogenetic females give support to a phylogenetic relationships between them. This group is endemic of the Neotropics. Further studies are needed to improve our knowledge on other Neotropic Macrothricids. Here you can see the references used in this presentation. Here you can see the publication of this study, and if you have any question, I will be happy to answer them. Thank you for listening to me.